Yeah, so this is another one. It's a EZH2 inhibitor. Actually, maybe we should do this first. Okay, so we should do this first. For prostate cancer, you guys remember homework 4? We look at androgen receptor. 80% of the prostate cancer in men are caused by an overactive androgen receptor. So androgen receptor is a transcription factor normally targeted or normally activated by the androgen hormone. For some reason, either it can be overexpressed or maybe the androgen hormone level is too high. The androgen receptor activity can get activated. And so very often, prostate cancer are driven by an active androgen receptor. And so the very early patients, you can just do surgery, take out the tumor, it's fine. But if the cancer cell has metastasized to other part of the body, we would not do surgery, instead treat the patient with a targeted therapy. The most effective one is androgen receptor antagonist, which looks like an androgen hormone. It will bind to the androgen receptor, but block its effect, right? So it's nuclear receptor inhibitors. And so they bind to AR, but block AR function. However, these type of drug, if the patient already have a metastatic tumor, sometimes after the patient were treated with the inhibitor for a few years, they become hormone independent. The receptor just can work without the hormone anymore. So previously, the drug can block AR function, and so AR doesn't work. But after, if the patient were treated for a few years, they still require the androgen receptor, but then it doesn't matter whether the drug is there blocking it or not. That's called hormone independent. And so this is like you can see here, like when people, actually we did a CRISPR screen of the cancer cells. So prostate cancer cells initially sensitive to the hormone treatment, and later on resistant to the hormone treatment. You can also try this by treating the cancer cell, with hormone or without hormone, and ask whether there are genes that have differential essentiality. And so you can see here, with hormone, there are let's see, some genes that are important, but without hormone, so you can see for the same genes that we are listing in here, with hormone, these genes are not so important in terms of, this is the p-value of the CRISPR selection, and this is the ranking of those genes. So initially with hormone, these genes are not ranked very high, but if you don't have hormone, this is, like when the cell are not treated with hormone, it's like treating the cell with a hormone inhibitor in the tumor. Right? So because in our body, you can't say no hormone unless you take out the prostate of the patient. But you can also treat the patient with, so you can do castration. So the patient will also not develop prostate cancer, but also sometimes after a few years, you also have patients with hormone, like a castration-resistant prostate cancer, which means they will not have the hormone, and the tumor will still grow. And so in this case, we're treating the cell, like normally it's with the, with hormone, but without hormone, you're asking, how can the cell still grow if you have like a drug or if they don't have hormone anymore? And you can see some of these genes creep up. Can you guess what the genes are on the very top that are significant in both? Can you guess what are the genes on the top in both? No, the genes that are top on both are the boring housekeeping genes, like ribosomal genes that you can't really target anyways. The genes that are really interesting is, if you don't use the drug, the cell can grow very well. But then if you give it a hormone inhibitor or you do castration resistance, castration to stop the hormone, and then this gene become really important. These are actually potential drug target. And you can see from this, we saw two genes, right? EZH2 and the BRD4. And so we ask, what is the EZH2? So EZH2 is an epigenetic enzyme. It interacts with two other enzymes in the genome called EED and SUD12. And this is a complex. They go to the genome to add H3K27 trimethylation. onto some proteins, especially histone proteins. Can you remember from homework 5, what does H3K27 trimethylation do? Upregulate gene expression or downregulate gene expression? H3K27 trimethylation normally downregulate gene expression. It's a suppressive histone mark. And so if you have a EED, EZH2 overexpression, the K27 is very, very high, then the nearby genes are silent. But without the H3K27 methylation at the promoter, this gene will be turned on. This is what people understand in most of the cancers, okay? Especially in some other cancer types. But so when we did the CRISPR screen, we found 2 hits, right? One is this EZH2, the other is BRD4. I don't remember, I don't know whether you guys remember, BRD4 is a gene that goes to the promoter of a lot of genes, and there's an inhibitor called the JQ1, which really goes to the BRD4 and target the super enhancer genes. Do you guys remember super enhancer in the epigenetic lecture? So in some genes, there you see a very long and tall regions of H3K27 acetylation or BRD4. Those regions somehow become super active. And in cancers, a lot of these peaks, their nearby genes, become super essential to the cancer. And if you can target them, actually it's very, very effective. And so there is a drug called the JQ1, which was developed by Jay Branner at Dana-Farber Cancer Institute. And he's now actually leading the Novartis, the whole oncology research. So with JQ1 is interesting is this is an inhibitor against BRD4. It also actually works on leukemia, but it also works on solid tumor potentially as well, which is, you basically, you can imagine BRD4 as a glue between promoter and enhancer. And if you have a super enhancer, then this nearby gene will be activated a lot. And then when you have a JQ1, it's like you cut out the glue between the promoter and enhancer. A lot of these genes will crash. The genes that depends on the super enhancers to turn on, will be much, much down-regulated. And very often, that will just kill the cancer cells. And apparently in this castration-resistant prostate cancer, BRD4 becomes super important. Another case is EZH2. And so actually, so we mentioned previously, EZH2 traditionally is known to be responsible for H3K27 trimethylation and suppressing gene expression. However, in the castration or androgen-independent prostate cancer versus the androgen-dependent prostate cancer, if we look at those genes that are treated with EZH2 inhibitor, we see kind of, or at least if you look at the genes that are different, a lot of these genes seem to be actually getting activated by EZH2 inhibitor, which is really strange. Like if you either knock out the EZH2 or treat the cell with EZH2 inhibitor. So normally if EZH2 is suppressing gene expression and you treat the cell with EZH2 inhibitor, do you expect those genes to be upregulated or downregulated? So if EZH2 suppress gene expression, and when you treat the cell with a EZH2 inhibitor, you expect those genes to go up, right? However, in the prostate cancer, when the cancer becomes hormone independent or castration resistant, when we look at those genes, you treat with EZH2 inhibitor, they become downregulated. It's very, very strange. And when we look at their actually, what are those genes? These genes are actually a lot related to cell cycle genes, and also those genes in a lot of different tumor, they are related to metastasis, worse survival, recurrence, deaths within 3 years or like really, really bad. And then we actually did a CRISPR study to see what's going on. It turns out EZH2 in a lot of the regions, indeed, they are bind to H3K27 acetylation. However, in the hormone-independent tumors, there is a group of genes that only has EZH2 binding, but they have no H3K27 acetylation at all. And these regions turn out EZH2 now is doing something else. It seems to be that it's kind of methylating some other transcription factors. And when we look at what those peaks are, these are actual androgen receptor peaks. So androgen receptor can become activated. Yeah, so if you look at the motif, where those, what those peaks are, these are the genes that really become activated in the hormone-independent prostate cancers. It seems like EZH2 is binding to those regions, and when we look for the motif, we see, androgen receptor actually is there, and you can see those regions in these locations. Androgen receptor don't bind at all, but in these bottom regions, actually androgen receptor bind to a significant number of those genes, and they kind of also upregulated the cell cycle genes to make the gene work much, much worse. And so this is actually a different mechanism now. So most of the time, EZH2 is a suppressor. It adds H3K27 trimethylation to the promoter of genes to suppress the expression of those genes. However, sometimes when you have a EZH2 mutation, it can now interact with other transcription factors, say androgen receptor or other transcription factors, to activate gene expression for other genes. And so it might be just adding the methylation to androgen receptor or something, that then you will have a different function, right? So you can see a lot of these epigenetic marks is very complicated. It can have, either in this case, tumor suppressor and oncogene in different conditions, right? So when we use the inhibitor, we need to kind of understand what it is really doing. So this kind of came to the situation. In the previous one, we identified these two essentiality genes from a CRISPR screen. You can also use CRISPR screens, treat the cell with the drug or without the drug. And in this case, we are treating the cancer cells with we first add the CRISPR, and we can also treat the cancer cells with or without T cells, or say with some control T cells that don't recognize the cancer versus the T cells that specifically recognize mutations on the T cells. So in this case, you can also see what genes that needs to be mutated in order for the T cells to kill the cancer cells in a differential manner, right? So here we are comparing the cancer cells killed with some random T cells that doesn't recognize it. Basically, this is no killing, versus other in another condition where we treat the cancer cell with a specific T cell that just recognizes the cancer mutation, and we are asking what kind of genes are activated, or what kind of guide are negatively selected or positively selected. So those that are negatively selected, so these are the resistant genes. When you knock it out, they will make the cell more susceptible for T cell killing. Whereas these genes, they are essential genes. When you knock it out, they become activated. They become enriched, right? So from this. This is another study that we did with collaborator Kai Wuki Fennick at Dana-Farber. We found that there are some genes that are depleted after the cells are treated with the specific T cells. And so on the very top there is PD-L1. Do you guys remember what this is doing? So in the cancer cells, remember, if the cancer cell is able to upregulate PD-L1, it's like giving the alcohol to the T cells to drink. And if you can somehow knock out the bottle, or it's like, forget it, like you do not allow the cell to express PD-L1, yes, the T cell can kill the cancer cells better. which means that it actually is a good validation that PD-L1 inhibitor should work, right? Because you're blocking the alcohol in some sense. And interestingly, there is another case. We saw 3 genes labeled in red in here. These 3 genes belong to the same chromatin remodeling complex. They actually help move the nucleosome around the different locations. Remember, nucleosome on the chromatin are like pearls on the necklace. So the chromatin remodelers help determine where the nucleosome should be placed. And interestingly, in this case, all 3 members of the chromatin remodeling complex get selected. And we can also validate, indeed, if in the cancer cells you knock out these 3 genes, the T cells can kill the cancer cells much, much better. And so, which kind of indicate that epigenetic inhibitors can be important, which we have seen repeatedly. If you use CRISPR screens comparing the cells with or without the drug treatment, very often you can identify epigenetic regulators as differentially essential, which means that they are very important players modulating drug response. It seems to suggest that a lot of these epigenetic regulators can control the cell plasticity. So every cell has a way of kind of shifting between one state to the other state. And these are all, so every cell remember, okay, I'm an immune cell, I'm a breast epithelial cell, or I'm some bone, some whatever cells. And so every time the cell is under stress, it's going to try to change its epigenetic status. Whenever it's under stimulation, differentiation signals, or under drug treatment or stress, epigenetic regulators are very, very important to control the cell plasticity for the cell to move from one state to the other. So it's actually very, very important for drug resistance, which suggests that they can be potentially used as a combination therapy. If you treat the cell with a drug, you do CRISPR screen, you identify what other epigenetic regulators are important. Those can be important additional drugs to treat the cell so that the cell cannot move away from that original